Namaste students, Khushroo I, JB and Dr. Chandrakala, Assistant Professor Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the lysosome. Lysosomes are the spherical organelles that contain the enzymes acid hydrolase. They break up the food so it is easier to digest. They are found in the animal cells, while in yeast and plant, the same roles are performed by the lytic vacuoles. The lysosomes are discovered by the Belgian cytologist, that is the Christian D. Duke, in 1955 and named as a suicidal bag or a suicide sac. The origin may be lysosome may be originated from the endoplasmic reticulum and the Golgi membranes. From the process of budding of prokaryotes do not have a lysosome, but some hydrolase have been located outside the cell. So the lysosome found in the higher animals. Lysosomes are the globular range in diameter from 0.2 to 0.8 microns. Under the electron microscope, lysosome appears dense and finely granules. Lysosome is essentially a pair of lipoproteins or a membrane containing enzymes. The membrane insulates the enzymes from the rest of the cells. If the enzymes are released, they can digest the cell, and hence the lysosomes are sometimes called suicide bag. Lysosomal membrane or a lysosomes are bounded by a single lipoprotein membrane. The membrane are impermeable to substances of the substrate of the enzyme containing in the lysosome. So here certain substance labilize causing the instability of the lysosomal membrane, leading to the release of the enzyme from the lysosome. The substance that stabilizes have a stabilizing action on a membrane. That is the labilizer here is the vitamin A, vitamin B, vitamin uh, K and vitamin E. The stabilizing examples are the cholesterol, cortisols, etc. The molecular weight of a molecule structures plays an important role as a part of the permeability of the lysosome membrane. The compound have a molecular weight higher than 200 and do not be diffused into the membrane. So here the lysosome which fuse form of the secondary lysosome and a primary lysosome. Here so lysosomes contain in this type of enzymes that nuclease which includes the ribonuclease and deoxyribonuclease, breakdown of the polynucleotide into a nitrogen base or a phosphate and a pentose sugar. Phosphatase, where the acid phosphatase break down the phosphatase ester to the monophosphatase. The lipase, where the lipid break down into a fragments. Protease, that protein break down into a constitute of amino acid. Glycosidase, where the beta glycosidase or a beta gluconase or alpha manniosidase break down into the polysaccharide to a monosaccharide. Sulfidase act as the sulfide ester breaking down into the fragments. The lysosome shows a polymorphism in different cell types and even within the single cell. Previously there are two types of lysosomes like primary and secondary lysosomes. Later it was three types like phagosomes, residual bodies and autophagovacuoles. So the primary lysosome is formed from an endoplasmic reticulum and a Golgi complex. The enzymes of a primary lysosome are synthesized by a ribosome and it is secreted in the enzyme outside the cell or stolen in them. The phagosomes are the form of the phagocytosis or a pinocytosis of the foreign material. The phagosomes associated with the primary lysosomes and undergo the fusion to form the secondary lysosome. Residual body is the final particle containing the indigestible material. It is form of the dough when the digestion is incomplete. The autophagocytes, the vacuoles are autophagosomes and is the lysosome containing some part of the cell and in the process of digestion. Lysosomes serve several functions like extracellular digestion, intercellular digestion, hormone secretion, fertilization, autolysis, or chromosomal breakdown. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session. Thank you.